Hi everyone. So yeah, I'll continue on some Mesoplasma florum science here. I'll present you an approach for decoding gene essentiality in M florum via Tn-Seq and hierarchical clustering. So my name is Simon Jeanneau. I'm a PhD candidate at Professor Pierre-Étienne Jacques and Sébastien Rodrigue laboratories here at the University of Sherbrooke. So the scope of my project, I talked about Tn-Seq and Anthony introduced me very well. He talks about transcriptomic. We are doing a lot of efforts to eventually drive integrative genome reduction rationally, so with the data driven. And my project is part of this whole large project, but I'm talking today only about transcriptomics, so only gene essentiality. Which is, yes, a part of this large project that was allowed by my two dear colleagues, Anthony and Dominique, that made a lot of wet lab work upstream of this. I'm working exclusively in bioinformatics, so I will show you more analysis stuff than wet lab technical details. So for a quick refresh on what is Tn-Seq or transposomic, but here we call it high density transposon mutagenesis because it's fun to make acronyms, but really we have a more specialized approach that is designed especially for our needs in the labs with Mesoplasma florum, for instance. But broadly, Tn-Seq is only true transposon insertion in genome randomly and once per cell, and and put it in a growth condition, selective, that will select for the transposon insertion, of course, and every cell that survive this that survive this growth with an insertion will be considered to be inserted in a gene that is not essential. So we sequence this insertion in the genome and can assess where there is allowed to be a huge insertion in the genome. So where the regions are not essential. So we call it high density transposon mutagenesis because we have quite highest density than before. We're talking about one insertion per about 30 cells, 30 base pair, sorry, in average. So it's quite high density. But I'll show you more details. But before, how we do our insertion is a very interesting way that has been that has a lot of optimization upstream. It's a PEG transformation that leads to the fusion of cells and with a suicide vector that transport the insertion vector. The insertion cassette that is inserted once in the genome, and the plasmid is no longer replicated or does not lead to multiple insertions. So it's quite a nice technique to have, and we're using for details purpose TN4001 transposals. But that's the technicals wet lab is done by now. I'll talk only about of bioinformatics from now. So what does this look like? Those data once they are treated a little bit on a track hub like this one, it's essentially lines on tracks. So as you can see, each line here, each tracks design a sample in a given condition where a vertical line represent one site where there can be an insertion at this time. So on the y-axis, you see a load of multiple samples that are all in the same condition. Here we are talking about semi-defined medium that Anthony talked about earlier, but with only glucose as a carbon source. And we are doing this experiment on multiple time points. So here we have a streak of 6 passage where we can observe the insertion degree or the insertion ratio at every time. That's very interesting. This visual inspection leads us to a lot of insights, even without doing nice and numeric approach. And only visually we can see that, for instance, the gene PEG46 is extremely essential because it's exempt of insertion almost totally. So at all time in the experiment, this gene was not allowed to have an insertion in, so it's strongly essential. And since we have a very high density of insertion, we can even uh, inspect the genes at a lower scale, so at a higher resolution. So here we see that PEG47, for instance, have a domain that is quite essential, even if the rest of the protein of the gene is probably not that essential because it's crippled by insertion. And on the other hand, we see genes that are extremely inserted. So at time zero, shortly after the insertion, we have a lot of after the metagenesis, I mean, we have a lot of insertion in the gene, and after some time, we can see that there is still a lot of insertion. So this gene being interrupted is not a huge deal for the cell with glucose, at least. Uh, However, we see a lot of cases that are uh, otherwise interesting uh, because like the peg 45, for instance, we can see uh, multiple things, but first a domain that is quite not necessary in the gene, so in the 5' extremity, but at the other end it seems to become very important, so it's exempt of insertion. And over time, the population tends to exclude the cells that have insertion in this gene specifically. So we could say that this gene, compared to, for instance, PEG49, loses its insertion faster, though, so the cost on the fitness of these mutants is higher. So with our time course, we could extrapolate a fitness score for each gene, and eventually we'll talk about reduction. Uh, so yeah, the density I talked about. So how to turn this into essentiality status? Because ultimately we want to say, can I remove the gene PEG45? And can I remove the PEG49, for instance? So usually this experiment of Tn-Seq of any kind leads to a density, a distribution of insertion, which is called insertion ratio. So when we look at the distribution of insertion ratio, we see that the genes are separated in mostly two population. For instance, these are E. coli data from our lab as well, where we see a peak, a super clear peak in close to zero, where all the genes that are essential are clustered. But we see a huge gamma fit here that represents a second population that are genes that seems to tolerate a certain level of insertion. But E. coli is it's very complicated compared to our minimal cell, quasi-minimal cell, the Mesoplasma. So here we have a very small population of genes, less than 800. So we cannot expect for to have as much genes that can tolerate insertion. Mesoplasma is a lot less versatile than E. coli, for instance. So most of the genes are uh, quite essential, and there is not enough genes that tolerate insertion to form a gamma fit population. So we cannot use the classic tools to separate our two populations statistically and be confident about where is the line between the two. So we had to make some curation work to create a hard essential goal set. So we isolated 61 genes that are mostly used for ribosomal proteins and polymerase and central carbon metabolism, and that we are strongly confident that they are essential. And we also validated with HDTM that they are exempt of insertion. So these genes, we are looking at their behavior, and we're trying to see what other genes behave like these, and can they be essential as well. We see this, these clusters, this group, only at even at a high level point of view with a basic clustering for visualization purpose. We see the lighter, the less essential it is because it's highly inserted. So we see that there's group of genes that are highly inserted, and there's group of genes that are highly inserted but less and less for the passage. But there is also a huge cluster that represents almost the half of the genome, even more in some case, where the genes are mostly not inserted. So I was wondering if there is there a way to mathematically isolate these clusters with a method that is robust and that is not arbitrary, because I don't want to set a threshold that is too hard. I want the analysis to be a little naive, let's say, by integrating all the passage. So I have a data for every time point for each genes. Let's merge everything together and see what the unsupervised learning can tell us. So I've designed this hierarchical clustering similar to what we see in the precedent slide, but I'm trying to isolate this cluster. So we're doing a hierarchical clustering with well, I will spare you the details, but there's a lot of parameters with this analysis, and I'm not done setting them to the exact optimal states. So this is all to take with a grain of salt. But when we go down the tree, we try to find the cluster where we still have the gold set within it, but when we go lower and in another branch, we lose the gold set, so we stop there. And all the genes that are in these clusters with the gold set are to be essentially. So so we isolate these co-clusters and deduce the essential status from them. With this method, I isolated a certain amount of genes depending on the condition. So Anthony told you there was semi-defined ATCC and multiple sugar isolated. So we find about 400 to 600 essential genes. So that's quite a large range, but that's not that surprising considering that most of the genes should be essential in Mesoplasma, which is almost already minimal. But we still have like 20 to 40% of genes that could be removed according to these numbers. So we are at the very beginning of this approach, but still, I checked out what were the different essential gene in some of the sets for curiosity purposes, and we see those that there is like a lot of genes essential in the two medium, for instance, ATCC and Similarity, but there is quite a shift in essentiality with Similarity. That is a medium considered more stringent, let's say. The stress level on the cells is higher in this medium, as we see like we saw the consequence on the growth rate. So we expect the cells to require more machinery, maybe, or well, more imports at least to survive in this medium. But all of these genes that are common to the let's say the core, let's say the 400 that consists the core, are mostly coherent with the literature. So we have a lot of genes that are I compared these genes with SYN3A and GCL. GCL here means some work that were made in our lab a couple of time ago. It consists of a GEM, so a model metabolic. Yes, so predictive. But well, what I want to see is that a lot of our genes are common to these models, so the literature and the previous work. So it's quite comforting when we look at this. when it's essential in both medium, it's also essential according to GCL, and it's present in SYN3A. But there is a couple of genes that are there is discrepancies about for these list of genes, and we have a couple of genes that are only essential in our condition and not at all in the models or in the literature. So there is a lot of investigation to lead at this point, and to recall the works on the sugar, I checked out quickly what was the terms that I found when I checked the quite sparse annotation of Mesoplasma florum, and I can see that, for instance, the PTS system essential in trialose that is not essential in fructose. The interesting point here, Anthony touched it earlier, is that we call this gene glucose specific. I would put money on the fact that it is not glucose specific. It is essential for glucose, of course. I checked out, but it is not only for glucose. So maybe there is some annotation to rework there with the condition we isolated. So maybe some genes in there can be attributed to some broad function at least, because there's a lot of these genes on either side and in the whole genome that has only hypothetical proteins. So this is the let's say the challenge with Mesoplasma florum, because the annotation is quite sparse. We have a third of the genome that is hypothetic, or so we expect to add some insight to the whole annotation at this point. So yes, there's a lot of investigation going on with these data. So I hope to provide some guidelines for reduction by providing the list of genes that can be clearly removed, and as well the one that can be removed but with a cost. So the genes that would have a fitness score that is to be evaluated depending on the chassis that is required at the end of the reduction. Well, push the annotation, as I said earlier, and it can be leveraged with transcriptomic data that Anthony presented earlier. So we have a lot of data that is boiling right now, and we have to cook something coherent with all of this. And I hope that transcriptomic and transposomic can be compared intelligibly, so it can be leveraged. Okay, so that's it. So that's the challenge I told. We have to guide our insight with data, because the annotation at the baseline is quite minimal as well. So I'll end here. So I acknowledge my two teams in which I work, and especially Anthony and Dominique that works a lot on this project as well. So thank you.
Well, it's clear who cares more about sterile technique or not. So Simon, if you were doing this, thinking about this is something that we have done also. I was introduced to a new transposon technology, the Tn-Seq or Tn-Seq system by Maria-Luc Senard and her student, where they have you do a higher density transposon bombardment, and then let Cre recombinase slice out the pieces that you slice out non-essential pieces. And I'm increasingly thinking that that's a better way to go. Is this something you've thought about?
Not yet. No, I haven't heard of this. How do you call it already?
So it drops LOX P sites into the genome with a suicide plasmid that makes Cre recombinase.
Yes.
And then it will lop out pieces of the genome. And so you get a population of cells where you've taken out the largest clusters of genes that you can do. And it gets you away from polar effects and quickly gets you down to cells. Again, it will take out clusters of cells. I mean, clusters of genes.
Mm-hmm.
I will, I don't know that they've published this, but it's in a chapter that they're writing with me. And they have shown me a manuscript, which I guess has never been published. I will ask Maria if I can share this with the community because I
Sure,
think it might be very useful for
of
you.
course. Yes, it would.
Um,
Thanks.
and then what is this, as you look at our transposon data and your transposon data, is it offering clues about annotation that maybe you, for us and for you?
Yeah, at this point, I haven't dived into deep annotation, re-annotation, let's say,
Mhm,
but we found some of the list of essential genes that were basically unknown. So uh,
mhm,
those couple of genes are only essential in CMRLAT with 3 allos, for instance, but they
mhm,
are unknown. I guess we would have a early insight on what this gene can
mhm.
are used to. They are at some point in the import or metabolism of carbohydrate, but I could not say how to pin out the very specific function of these genes at this point with Tn-Seq.
And why did you choose TN 4001 as opposed to TN5? I mean, we tried both.
Interesting. Those questions were addressed by my colleagues some times ago, so I would be. I don't know exactly the reason, the technical reason behind it, but I'm confident that these colleagues took the same decision. Maybe some people of the Rodrigue lab could answer the question
Mhm. I mean,
because
it could be as simple as what you had in the freezer. Um,
true.
okay. Other questions for Simon?
Yeah, I have it's Troy. Thank you for the talk. I just have a follow-up question kind of related to John's second question. Not necessarily with respect to genes that you know that they're there, but you don't know the identity of them. Have
Hmm.
you been able to see sites of like high levels of bombardment that are not annotated as any genomic feature? And the idea, or why I'm asking this, is those could potentially be like regulatory elements such as like riboswitches or things like that that you are not, they have not been annotated yet, so they are not unknown, or they're not characterized as unknown proteins. They're just completely unknown
Hmm.
to even exist on the genome, to
Yeah,
phrase it that
that,
way.
that's a
Troy, do
very
you mean Troy, do you mean high levels of bombardment or high levels of no bombardment?
I guess, yeah, it would be high levels of no bombardment
yeah.
in regions. Yes, yeah. Thank you, John. In regions where there's essentially no gene, so you would expect that there could be bombardment there.
That's a very interesting question. I'm very interested in all the non-genomic, non-genic features in Mesoplasma, but there is not a lot. I mean, the intergenic region in Mesoplasma florum represent like, if I remember correctly, less than 10% of the genome. So there is not a lot of space for features that are regulatory, that are imported for regulation and that do not overlap with genes. So
Mm-hmm.
uh, I've been looking at this visually, but we did not lead investigation on this front yet. But I found some very small bins between genes that appears to be essential in certain condition. Well, those are not shown here, but I was wondering at this time, have I missed some annotation because it's barely possible? But you have a good point. Could be some regulatory non-coding features like robust switches uh, or any non-coding RNA, for instance. But I'm staying aware of this because it could be, of course, some interesting insight with our density. We couldn't find it.
Okay.
John,
Yes.
coming back to your question, in fact, we tried both. We tried TN5 and TN 4001 as well,
Mhm.
and we got better results with TN 4001. That's the main reason.
Interesting. We got the same. We got that with genitalium, but not with mycoides.
So, yeah,
and
that's
and Alligatorus was exactly the same. But we also saw that there were genes that were hit with one system that wouldn't be hit with the other. Thank you,
Good.
Dominic.